Hi there, welcome to another episode of Katie the Science Lady. I'm Mrs. Jacobson, and today's topic is non-Mendelian traits, incomplete dominance, and codominance. So let's learn together. All right. Today we're tackling non-Mendelian genetics, and I've split it up into two parts to make these a little bit shorter and easier to digest. So today we're tackling part one, which is going to take care of our incomplete dominance and our codominant examples. The first thing we're going to do is a quick recap of complete dominance, or just regular Mendelian dominance, so typical stuff here. This is where one allele completely covers the other. That means it's dominant if it's going to mask the other. It's recessive if it gets hidden. We use uppercase and lowercase versions of the exact same letter, so little b, big B, that kind of jargon. And it would look like this in a Punnett square. We've got a big P here, a little p here, big P here, little p here. This means we'll end up with 1/4 of the offspring being big P, homozygous dominant, 1/2 of the offspring being big P, or heterozygous, and 1/4 of the offspring being little p, or homozygous recessive. And we get different colors here. We have distinct different phenotypes or physical appearances. We have those that are dominant phenotype showing up purple, and those that are the recessive phenotype showing up white. So we get two specifically different colors here. Now let's kind of mix it up a little. Incomplete dominance is different. Obviously, there's a different name. Neither allele is dominant in this case. So when we have a heterozygous offspring, they're going to have a blended phenotype. So imagine you took two colors of paint and then you just poured them together and you mixed it up really, really well. It would be a blend of whatever colors you put in there in the first place. And that's what happens with our heterozygote. We use different lowercase letters for incomplete dominance. I tend to use uppercase letters because it just helps my brain, but lowercase letters are usually the way to go. If we take a red flower, which could be little r, and we cross it with an all-white flower, which would be little w, we're going to get a pink flower if it's heterozygous, which in the case of this Punnett square, you're going to get all pink flowers here. So they're going to be all little r, little w, which will be pink flowers. The key here is that it's a blended phenotype. You can use uppercase letters if you want. It doesn't really make a difference. As long as you know that it's incomplete dominance, how the heterozygous offspring kind of react to it is all you need to know. So it's up to you how you want to notate these. Typically, they are shown in lowercase. I tend to use uppercase because it's just kind of the way I work. But as long as you know incomplete makes a blended phenotype, you're good to go. Codominance, on the other hand, is, again, completely different. Both alleles are dominant here, so we have no recessive alleles. So that means our heterozygotes are going to show a mixed or patterned phenotype, aka they're going to show them both at the same time. So you're not going to mix together, you're going to just see both of them. Like if you took two kinds of paint and you splattered it on the wall, you're going to get a random pattern of both colors. And that would be codominance, because co means together. So they're together dominant. We use different uppercase letters for these, and again, I always use different uppercase letters, just kind of how I roll. So if we mix this all black rooster here with this all white chicken here, and we gave them the letters big B for black feathers and big W for white feathers, and we mix them together, all of our chicken offspring are going to be big B, big W, or BW. That's going to give us a checkered chicken. That is what they're called. And yes, I actually have seen them. My family used to raise chickens, so they are real. I'm not lying to you. And this is what they look like. They're pretty cool. So it looks kind of like a stripe, but we do call them checkered. These phenotypes of the heterozygous offspring might appear striped or speckled. You may have different patterns going on, like this checkered appearance, but you'll get a mix of these colors. You'll see them both showing up on as the phenotype. And again, that was big B, big W, one of each allele because they're heterozygote. And again, you'll see it like this in the flowers. They may look like they're tie-dyed or kind of watercolored. They're really cool looking. And codominance is pretty prevalent in nature. So I'm sure you could go out on a nature walk and find an example of codominance if you tried hard enough.
Let's recap what we've learned today about codominance and incomplete dominance. When a trait is codominant, it means that both alleles are dominant. So you're going to see both of the phenotypes in the heterozygote. This could take a couple of different forms. They could be checkered, like in this checkered chicken. They could be striped or speckled, like a lot of the flowers you may see. You may see flowers that look like this, that are kind of red and white striped, but they can be any number of colors. If a trait has incomplete dominance, it means that neither allele is dominant over the other. This means that when a plant or an animal is heterozygous for the trait, they're going to show a blended phenotype, or a combination of both. So a lot of times this just kind of takes our brain naturally to a place where it would go. If you have a very dark purple flower and a very light white flower, you would naturally think that their offspring would be a light purple, kind of a lilac color. And that's the case here. If you take a red flower and a white flower and it's a heterozygous offspring, it will be pink. Please click here to watch some of my example videos from my How to Genetics series. And as always, I hope you had fun, I hope you learned something, and I'll see you later.